So I'm Jim Knierim. On the Zoom, and they have Jeff's name underneath me. But if you like to talk, I'm Jim. If you don't like it, I'm Jeff. So, um, so today I'm going to talk about some of the work that we've been doing about the path integration system. For those of you who are, so we're still cut off over there. I tried to shift everything over. But anyway, the notion of the path integration system is that uh, is the notion that as an animal moves through the world, from time t to t1, there's a vector of movement, a direction and distance. And in order for that to work in the brain, there has to be some kind of mapping between the distance and direction traveled in the real world to the update of the position in the animal's cognitive map. So this is just a little sheet of tractor cartoon of a bump of activity that has to move from one place to the other in sync with the animal's uh, change in position. If the question we are addressing is how does the brain know how much to move the bump in the cognitive map relative to the animal's movement in the world? And we call this the gain factor of the path integration system, the G, that and by definition, G has to equal 1. You know, that if the rat moves a certain displacement in the world and G is 0.5, well, this bump of activity is only going to move half the distance. And as the animal moves around, clearly the representation in the brain of where the animal is going to be dissociated, decoupled from where the animal really is in the world. So the question we have is that how does the brain learn this G? All models of path integration just assume that it's G, that the update in the brain will faithfully reproduce the animal movement in the world. But we think this must be something that has actually learned through feedback with visual information. So in order to test that, we developed this apparatus we call the dome. I can't really see how many people, what you can see over there, but on the left of the folks who did all this work, Manu Madhav, Ravi Jayakumar, Francesca Savelli, and Noah Cowan, who just arrived, has been a collaborator for many years now. We've done some great work together. What the dome is, it's a virtual reality apparatus in which the animal actually physically moves through the world. He's tethered to a boom arm. Here's what it looks like from outside. We take this hemispherical shell, we bring it down, which is opaque. Here it's just rendered transparent so you can see inside the dome. We project cues inside the interior surface of the dome, and these cues are rotated as an array, as a feedback function of the animal's movement through the world. So as the animal moves around, say in a counterclockwise fashion, we can rotate the cues inside the dome either in the same direction as the animal or a different direction to give the animal illusions of running faster or slower than it really is. And that's shown in this cartoon here. Let me go back to not the laser pointer. You can see here, this is what we call a gain greater than 1. You can see as the animal moves around the world, the landmarks are rotating the opposite direction. Let me do that again. And you can see we're going to plot data in the lab frame. This is the actual animal's actual movement through the world. But relative to the landmarks, you can see the animal's running twice as fast. We can also make a gain less than 1. In this case, the landmarks are moving in the same direction as the animal. And you can see as the animal moves in the laboratory framework, it moves a certain distance, but relative to the landmarks, it's actually moving slower. So by using these gain factors, having the landmarks move in the same direction as the animal at different speeds, or in the opposite direction, we can give the rat the illusion it's moving faster or slower. And these are the kind of experiments that we've been doing. What I'm plotting here is the activity of a hippocampal CA1 place cell. Laps are on this axis here. Each gray and white bar is one lap. And plotting here is the location as the rat runs a 360-degree lap. We start the experiment at a gain of 1. That means the landmarks are stationary. They're not moving. And as for the first 7 or 8 laps, the landmarks are stationary. And you can see here's a place field that fires when the rat runs down the track. It starts to fire at this 270-degree location on the track. And every time the rat passes that location, the cell fires. It's just a place cell. Now, at this time, we start to ramp up the gain of these landmarks, moving as they're moving in the opposite direction, but faster and faster. And you can see that the place fields start to drift around until we reach our target of 1.8, and then we maintain that for a number of laps. So by now, in the lab framework, the landmarks are drifting around. But if you plot the firing of the place fields relative to the rotating cues, you can see it's very stable. This is classic visual landmark control over the firing of these place fields. The question we have is, what happens at this point when we turn the landmarks off? Now the system is only under the path integration, the use of the animal's self-movement to keep track of where it is. Under normal conditions, if we had done that after a gain of 1, the place fields would more or less stay also firing once per lap. But now we've given the rat 30 laps of experience in this altered reality, where it actually is moving twice as fast as it thinks it is, covering twice the distance. The question is, when we turn the landmarks off, does the system revert back to firing more like once per lap like it did here? Or have we recalibrated the system so that it continues to fire in the landmark frame as if the landmarks were still there, rotating around? So maybe firing twice per lap or 3 times per lap. And the answer is sort of in between, but clearly what you can see in the lab frame of reference, the place fields are still firing without really any spatial tuning relative to the world. But in the landmark frame of reference, they're firing in a much more consistent way. There's some drift that occurs lap by lap. But you know, if this is every lap, the fields are only drifting a little bit. So we basically recalibrated how far the rat thinks it's moving with each step as a result of the training we've given us in this landmark virtual reality. And what's shown here is the gold line here is our measure of of how fast the system updates as the animal moves around. And you can see that it's actually overlays on top of the experimental gain, showing the gain control. And when the landmarks are off, the system pretty much fires as if the landmarks were still there. So we call this path integration gain recalibration. We trained the path integration system to act as if the rat is still in the virtual reality with being controlled by the landmarks. And to show this is not just one example, this is the data we published this a number of years ago now. For the different experimental gains we gave, greater than 1 or less than 1, you can see that we always recalibrated the path integrator when we turn the landmarks off in the same direction as what the landmarks had done. Each color is a different animal, different experiments. The dashed line here would be perfect recalibration, but what you can see is that typically the system did not recalibrate 100%. There was always some error left over. So what we're trying to do now is understand more of the dynamics of how the system works. And 2 questions we have is about the internal theta rhythm dynamics in the hippocampus. How does that contribute to this path integration gain recalibration? And I'm going to tell you 2 stories about theta. One done by Yotaro Suyoka, who's in here, going to give a talk in the next session about a different topic. And then talk about theta phase pre-session and pro-session. And Sungbum Park is a postdoc who's talking about theta, doing work on theta frequency as an indirect readout of the hidden value of the path integrator. So let me talk about Yotaro's work first. Theta phase precession is a phenomenon discovered by Okich and Recchi in the 1990s. The notion is as the hippocampus has this very prominent A-Hertz theta rhythm as the animal runs around the environment. And this modulates the firing of these cells. You can actually hear it on an audio monitor as the animal runs through a place field. That's 8 Hertz rhythm. What O'Keefe and Recchi discovered is that the firing of the cells shown by each of these tick marks here at each cycle of theta, it's not locked to the same phase of theta, but actually the phase of the theta rhythm that each burst occurs is earlier and earlier in the cycle. And that's what they call phase precession. And you can show that by looking at all the spikes of the cells on the dots here. This is the position of the rat as it runs through a place field. This is the theta phase. We're just showing it for 2 cycles of theta. You can see that there's this clear relationship that as a rat enters the field, he starts at a later phase of theta, and as the animal runs through the field, the phase precesses earlier and earlier. The question is, what happens in our apparatus with the dome? Now, As I showed you before, in the landmark frame of reference, as we're moving the landmarks around, the place fields are controlled by those landmarks. And if you plot out this cartoon place field in the landmark frame, when the landmarks are stationary, if the gain is greater than one or less than one, the place fields are more or less the same size relative to the landmarks. But what does that mean relative to the lab frame? What that means is when the gain is greater than one, basically the landmarks are moving in the opposite direction of the animal. If the gain is two, that means this place is going to fire twice per lap in the lab frame of reference. The whole map is going to be shrunk in half, which means the place field itself is going to be half the size in the laboratory frame of reference. When gain is less than one, now the place field, the landmarks are moving with the animal. That's actually going to stretch the whole map out. And each place field is going to be much larger than it is in the lab frame of reference. Now, the theta rhythm doesn't change much during these manipulations. So what this means is when the rat runs a g of 1, it goes through a certain number of cycles of theta. When g is greater than 1, the number of cycles of theta are much less. At g less than 1, the rat runs through the field, there are many more cycles of theta. The question is, how does the system react to that? What happens to this phase precession? And the answer is that in the landmark frame of reference, the theta phase precession is maintained. The system doesn't care about whether it's going to take two cycles of theta or seven cycles of theta or 25 cycles of theta to move through the field. It's going to precess by the same amount relative to this landmark frame of reference. So that's interesting. We didn't know what would happen. We had some ideas from the prior literature, but that's not too surprising given other experiments. But what Yotaro discovered as he looked at this more carefully is that there are interesting differences based on the precession lobe of the rhythm being bimodal. And this has been known for a long time that the phase precession is not really linear. It's actually two lobes. There's a lobe, as the rat runs through the field, it goes down here, but then it looks different down here, that this is actually a bimodal phenomenon. And work by Wang et al. From Brad Pfeiffer's lab showed a couple of years ago that actually this part of the phase precession plot is actually indeed this phase precession, as I mentioned before. But this lobe is actually phase procession. You don't see it really here in this diagram, but in their paper they show very clearly what's happening here is that the phases are advancing at each cycle of theta. So this is like a replay of what's going on, a prediction of where the animal is going to be. This is actually a recapitulation of a backwards shift of the animal's representation. So these two lobes show two different effects. One is phase precession, this is procession. Now, what Yotaro discovered in our experiments, when you had a low gain condition, this second lobe was much weaker or even disappeared. And in the high gain, it also was weaker. But when you had a middle gain close to one, the second lobe was there. So in our gain experiments, even though you see this phase precession is still there, we lose that second lobe of the phase precession plot when there's a mismatch between the animals moving through the world and what the landmarks are telling the animal to do. This is consistent with a number of models of theta phase precession, work by Mike Hasselmo and others, suggesting that this early phase of theta is actually when new learning takes place. And we think that's what's going on in our experiments. As the animal runs around and there's this mismatch between its self-movement and the landmarks, it has to continually learn these associations between, okay, now when I move this fast, I move this far. Oops, now I move this fast and I'm moving further. It's learning this new relationship between its movement to the world and how that reflects the landmarks. And we think that the fact that, according to Hasselmo's modeling, shutting down the spiking activity in this learning part of the theta cycle is necessary to prevent interference with old memories when you're storing new ones. Yotaro took a model from Chu and Ji's from Si Wu's lab, where they looked at theta phase precession. It's a ring attractor, a continuous attractor network, but what they added into it was this a spike frequency adaptation. And I won't go into the details of that model, but it's just what they've shown in this model is that when external input drives a bump of activity around this ring attractor, the spike frequency adaptation actually causes as the movement can go forward, but then it actually causes an oscillation back and forth around the input drive. And what they found is that depending on the strength of that feedback inhibition, they can actually cause this second lobe to appear or disappear. What Yotaro did was basically show, well, in our case, we can take that model and show a similar phenomenon if when the input from the allothetic cues, the landmarks matches that from path integration, you get a certain amount of input into the excitation. But when there's a mismatch between the allothetic cues and the idiothetic cues, that means there's less excitation into the ring. And we can actually cause the same phenomenon that when there's this mismatch, that actually produces the loss of the second lobe, but the first lobe is intact. I'm going to quickly then talk about the second part here, the work by Sung Bum, also looking at theta frequency. The question here is that when the landmarks are on, they are driving the place fields. But as I said before, we think the path integrator is still always active, but we can't measure what the gain of the path integration system is when the landmarks are actually overriding it, causing the place fields to fire relative to the landmarks. So how do we understand what the dynamics of the path integration gain is if that gain is being overridden and hidden by the influence of the landmarks? What Sung Bum discovered, also Manu Madhav first found this, is an interesting difference in the theta frequencies. So here's our experiment. As before, we start out with a gain of 1. There's an increase in the firing, the increase in the gain, and then we keep it stable. On this axis here is the frequency of the theta rhythm. What we find is that as this rat starts running and we start changing the gain, actually the frequency of theta decreases. Now, when we stop changing the gain, the frequency increases of theta. And here's another example of a situation where as we're changing the gain of the landmarks, theta frequency decreases. When we stop changing the gain and make it stable, it starts to increase again. And if you look at all the different experiments together and average them, you see that this is actually clear. We did all the statistics. This is highly significant. That when we start rotating the landmarks, that causes the decrease in the theta frequency. When we stop rotating them, keep them stable, the theta frequency starts to recover to its initial value. What Sung Bum then did is we plot here the change in theta frequency from the start of the experiment to when we turn the landmarks off. What's that difference? Plots that on this axis. On this axis, we plot how much is the difference between the path integrator gain. How much recalibration is there? And we see there's a clear relationship here. The amount of recalibration, how close this line is to the ideal, depends on how much the theta frequency recovered. He used the same model from Chu Ji et al. And shown that the frequency of this oscillation changes as well. I won't go into the details of that, but basically we were able to use the same model to show this same effect. So just to conclude, we've shown here that the theta phase precession is unaffected by the conflict between the path integration system and the landmark navigation system. The phase procession lobe is weakened. It's consistent with the models of early phase of theta being specific for new learning. Theta frequency is an indirect readout of the current state of the path integration gain, even when it's being masked by the landmarks. And this simple continuous attractor model modified from Chu and Ji et al. Can account for these. Noah Cowan's going to talk next session about modeling we've done on this, and with Yotaro's go to about head direction cells, and then a couple of posters, Bharat Krishnan, about other things we've done with the influence of landmarks onto these cells. So I'll stop there.
You have 40 seconds.
Um, what what happens to phase precession when the gain is zero or negative?
We haven't looked at that. We haven't done those experiments. When the gain is zero or negative, the rats get really freaked out when they do the experiment anyway. So we don't know.
So path integration, this is happening all the time. If we're moving through the world and animals are picking up speed or slowing down, path integration. I'd always kind of thought that basically the change in data is sort of not for the first time in person. But I was wondering if you think that it's adapted all the time in the natural world.
The question is, as the animal moves, speeds up and down, as it actually moves, does that change the path integration gain and so forth? And no, you're absolutely right. Path integration only works if, as the animal moves slower or faster, the activity in the hippocampus can take account for that. So the gain recalibration doesn't depend on the actual animal's actually movement. What we're showing here is that as the animal moves through space, whether it goes faster or slower, it's actually changing how far it's moving relative to the landmarks. And that's what's causing this system. The path integrator says like on this far, but relative to the landmark says that, well, I actually went further than that. And that's the error signal that then drives the recalibration of this thing.
So you can talk more about this during the break. Thank you.